Alright, now welcome to the next part of the video, which is on Specimen C, still on fowl, lungs from domestic fowl, freshly procured. Okay, freshly procured, just like the liver, the lungs, here is an organ of respiration. Okay, so the oxygenation function of the liver, of the lungs, rather, just like you know that this is an organ of respiration. This is the pathway, the air that you breathe contain oxygen. So the aspect in which oxygen is used in the process of oxidation of substances, food substances, taken during digestion, prior to release of energy. And in turn, the same organ also remove carbon dioxide produced. So these are the functions, executive function, removal of carbon dioxide is your accessory function. And also gaseous exchange. Yes, gaseous exchange is quite different from composition. This is more like internal. This one is more like external. Okay, this is just like the breathing in aspect of respiration, and then the taking out, breathing out aspect of respiration. It's making sure that the breathing is balanced. That the amount of oxygen that you take in, and then the amount of oxygen you take in from principle of diffusion, oxygen goes in from where it is much more to where it is less. Okay, sorry, where it is less, okay, to where from where it is small, from where it is more to where it is less. Okay, that is what it is. That's based on principle of diffusion. The carbon dioxide at that same point is also removed from where it is more produced in the cell. Okay, oxygen is taken from the environment, carbon dioxide is being produced in the cell and then taken out. So this is a gaseous exchange. Also, the lungs also store oxygen. Maybe in the time of need, oxygen is also used in some cells. Okay, so these are the functions. So let's look at the observable features. What I want you see when you are looking about the lungs of a bird. It has a unique shape. Okay, unique shape is that much unique compared to lungs of mammal. The uniqueness is why the lungs is well fitted for its function in birds. Secondly, it also has a thin wall. Thin wall is for efficiency of diffusion, just like any other respiratory surface. Okay, well adapted. And a thin wall. The bronchi, you see some air spaces where oxygen can freely go in. Bright red color, just like the liver. The only different is one is deep or dark reddish brown. This is bright red color. A spongy texture, not that smooth. Also, it's low flow or light weight. If you compare the weight of the lungs and that of the liver, if you are asked to compare Specimen B and Specimen C, you can use their weights. Okay, light weight, much weight. Comparing the weights plus spongy texture. So these are the comparison you can see. In this case, just like you do know that the liver have many lobes. In the case of bird, there are 2 or 4 lobes in the liver of a fowl. But here, for lungs of a fowl, there is no lobes or features. But also you do see lobes in the lungs of mammals, but not that of this. There's several air sac connections in the lungs. These are the things you're going to see, you're going to observe physically when you are experimenting on the structure, the lungs of a fowl. Okay, so these are basic points in hospital. And again, to also differentiate between the Specimen Specimen B and Specimen C. And whenever you are differentiating, you must tabulate. This is Specimen B, the former. This is Specimen C. Okay, if you are to state categorically the observable structural differences, what you see. Okay, starting with the color, Specimen B is kind of dark reddish or deep reddish brown. Okay, color. So what's the color of this one? Here is the color of this one, bright red color. Bright is much deep brown. So this is just like red. Okay. Secondly, you see in B, there are two main lobes. Okay. Position of lobes. Position of lobes. Here there is total absence of lobes. As you can see. Here J is not a sac. As far as liver is concerned, it's not an organ where air can go. So a sac, you can see a sac absent. A sac absent, as well as our local. A sac absent. Here a sac present. So whatever you see here, and if we compare it with the observable feature of that, this can also form part of the structural difference in specimen. Okay, let's also look at the structural. Okay, now and also this is very important weights. Okay, for this, for heavy weights, and then the lungs of the specimen C is also light. I don't think we do for structural differences. And to similarities, the color, the fact that the red appear in the color, so you can see both are red in color. As much as one has kind of touch of brown, the other one is just brown, but there's touch of that in that this. So both are also red in color. Okay, and you can just also look at it. It's just very minor. Okay, blood vessels are in both. So you can look at the color. Okay, in this specimen we're going to be looking at the whole gizzard from a domestic fowl. Whole gizzard from a domestic fowl. Okay, as the device is being without reason, whole gizzard from a domestic fowl. What are the observable features? There you see a muscular wall, a rough surface. That's what you see next. Next you see a keratinized lining which protects the muscular wall. A keratinized lining which gives protection to the muscular wall. Then you also see grit and stone parts. It's visible, sometimes it's not visible to the eye, apart from the stone that these fowls pick during scratching food from the earth surface. Then you now see gizzard teeth bearing down to the grit and stone. And you now see a connection of the gizzard to the small intestine. You see a connection, you see something that connects a gizzard to the small intestine. If you look at the picture diagram, you will see that in the video, something that connects the gizzard with the small intestine. So these are the things you observe. Carefully observe a gizzard of a fowl. You see all of this, a rough wall, a rough part, muscular wall, which is being protected. Sometimes when a fowl is killed, you can see that part that is being removed, removed before the fowl is prepared for meal. That is skeletonized. Letting you see it removed most of the time. You see great stones, you see gizzard teeth, you see a connection, something like that structure that connects that extension of the gizzard, which now connects to the small intestine. Now let's look at the function of the gizzard. Mechanical digestion. Yes, mechanical digestion. This is the parts, this function is the grinding, okay, the breaking down, the breakdown. So look at the parts that take part in breaking down. The gizzard teeth, the muscular and the grit and the stone. That helps in mechanical digestion. The food is stored temporarily in the gizzard. Okay, in readiness to crushing. So in readiness to grinding. Okay, but the mechanical force that grind this food when it has stored temporarily. So it's a result of food storage. So I'm mixing this digestive enzyme after the grinding. Okay. So the food is stored in the gizzard. But that is not permanent. It's temporary, waiting to be crushed. Okay, to do mechanical action. Okay, mixing after the crushing, after the mechanical digestion, the food, the grinded food is mixed up with digestive enzyme. More crushing also happen, crushing or grinding. This is where the food that the bird had already picked up together with stone. This is where the stone that the bird picked at the point of picking from the eggs, they do help to grind the food. Then also the gizzard also help to separate those food that were not able to digest. That's why most times if you kill a fowl, you will see some tiny pieces of stones, you see some grains. These are the part of food that were not able to digest. So the gizzard will also sort those ones out, those food that were not able to digest at that point. You also see regulation of food passage. Yes, the gizzard will ensure that those food that really are digested pass through the small intestine for further enzymatic action. Okay, for further enzymatic action and as well as absorption. So the gizzard will also sort out those ones that are not yet to be digested and those ones that are already digested. So those ones that are digested pass through the test, so they go through the next stage of digestion and absorption. So these are also really useful to have regulation of passage, regulation of food passage. Then also some nutrients also do absorb the gizzard. And again, it's also very, very essential for survival of the bird. So these are the functions of the gizzard. Not only gizzard for the basic part. There are some other organisms where you can also see the structure apart from the basic part. You can also see the structure in this organism, which is called earthworm. Earthworm is an invertebrate. The examiner may ask you mention one other organism that also, apart from this particular specimen, do also have gill. Okay, use gill for digestion. Gizzard is very, very important structure in there, which aid digestion, most often mechanical digestion. So if we want to compare this with the former, the Specimen C, which we just finished now, which is a lungs. There's so many things that you want me to see now. Based on what we already see as observable features, okay, connect it with that of Specimen C and see how this one differ from that one. Okay, and then the functions, how the functions also differ in this one. This one is an organ that aids digestion. Okay, this other organ, that is the lungs, is an organ that aids respiration. Okay, liver function is neither here nor here. I said liver can also aid some functions. Okay, so some digestive related functions, okay, and other functions. But this one specifically is a digestive function. So I'm not talking about the elementary tract of a fowl without the gizzard. The gizzard is just like the mouth. Since the mouth is being modified into a beak without teeth. So what they do use now is that the teeth do occur in the gizzard. That's why you can see gizzard teeth, which with the help of great stone, and those stones that also pick at the point of picking food from the earth. All of this help in crushing and grinding the food prior to digestion. As you can see in the video, where you always see all the structures I just mentioned, the keratinized part, you see it, okay, that's white in color. That part contains the muscular wall. That part is most likely removed because eating it is not digestible. It's not a digestive part. Okay, because keratin are protein that are fibrous in nature, so they don't easily digest. So that's why that part is most likely removed anytime when the bird is prepared for the part that you want to say how it has to we have gizzard teeth. We don't have the part of the gizzard that is connected to the small intestine, because the small intestine is the next portion of the elementary tract of the fowl, okay, that goes down to the last part of the elementary tract. So we're not talking about the gizzard. It linked up with the small intestine. So food that are temporarily stored in the gizzard are later discharged. Most especially those that are digested. Those that are not able to digest, those indigestible ones still remains in the gizzard for some time. Okay, next we're going to be looking at the next specimen, Specimen E. Alright, Specimen E, whole intestine from a domestic fowl. Still on domestic fowl. Observable feature, that will be the first question you will likely see. The series states at least 3 or 4 to 5 structural observable characteristics of the domestic fowl. You find that the intestine of the fowl is long and thin, very thin. Okay? Long and thin, and also coiled in shape. Okay? And also the walls are thin, and then also pinkish. So look at how it's called. Observable features 1, long and thin is called. 2, coiled shape is called. 3, thin walls is called. Pinkish color. The reason why the color is pinkish is because of blood vessels you see them passing through the intestine. And that gives it that pinkish color. And here there is no distinctive color, just you see in intestine of smaller parts like varieties color. There's no distinctive color as far as small intestine. You can't see that color there. Okay, you now see the cloacal connection. Cloacal marks the end of the small intestine. Okay, where you see the urinary part, the reproductive part, comes in there. That particular structure is in dual function. Okay, both the urinary part is there, the reproductive part is there, and as well the digestive part. So these are the cloacal. Maybe you may ask which of the parts, the structure of this particular specimen aids reproduction, where collection happens. It is the cloacal connection. These are the things that I am going to observe in Specimen E. So now let's look at the function of an intestine. Okay, the function of the intestine, just like the intestine of a fowl, the function of the intestine still remains same for every organism. First of all, absorption of nutrients. This is where nutrients, like all end product of food, okay, all digested food substance like glucose, amino acids, fatty acid, and glycerol, vitamins and minerals. All of this food the nutrients of this food that is already a nutrient, they're all absorbed in the small intestine. It's also responsible for absorption of water. The small intestine also eliminates waste products. Okay, this is also where there are some enzymes. Okay. Most enzymes that are produced in the small intestine are enzymes for carbohydrate digestion. Okay, enzymes for carbohydrate digestion, enzymes for proteins, enzymes for fats and oils. Okay, this is where you literally see the enzymes that act on this food. So you'll also see enzymes like maltase. Okay, enzymes like rhizome. This is where you see such enzymes. Other enzymes like lipase, enzymes like invertase, enzyme like lactase. These are where you see this enzyme. All these enzymes are responsible for catalyzing or breaking down this food substance. Okay, you see these enzymes in the small intestine. So production of digestive enzymes. Also storage of food, the food that are stored in there temporarily. So the key important point I speak from this particular specimen is the fact that question must come as far as this specimen is concerned. The examiner must ask a question on the structural observable features of the specimen. Look at it here, long this and the function of this specimen. Okay, now let's go to the next specimen, which is Specimen F. Okay, the specimen here is ginger. Okay, we have now leave everything and we call fowl. So we're looking at a food aroma, a ginger rhizome. Okay, so let's start with the observable features. Now, when examiners ask observable features, their point of view is to know if the candidate, the student, can be able to state the facts of what they can observe from the specimen, the live specimen. Okay? Yes. So what are the things you observe? You observe that the shape is irregular. Yes, you see the nodes and the internode here and there. That's what makes the shape irregular. And what is the color? The optic color of this ginger rhizome is brownish. Okay, it's brownish in color. Okay, the ones I know is brownish in color. This is what you see. If you see any color, you specify it. Okay, now you have the fibrous texture. You have a very rough surface that is fibrous structure. Just like mentioned, irregular shape, you see nodes and internodes here and there. If you look at the video, you see the nodes. Okay, you see the antinodes, nodes and antinodes. You also see that this rhizome ginger also is aromatic. Okay, it gives its particular aromatic odor to some. Some do like the color, some do not like the odor of a ginger. Okay, so it's also, you notice that you see some scars and marks all over this specimen. You see some natural scars, natural marks, you see some marks all over. So that's what they also carry on with advent teachers. So these are the things that is structurally observed when we are dealing with ginger in that exam. So what are the functions of ginger? Storage organ. So these are where minerals, where water is stored. Okay, vegetative propagation. Okay, that can disperse and start to grow just like that from its vegetative part. So the food source can also add as a food source. You can also use it as a food additive. Additive, you see flavors and aroma. Sometimes it's pleasant. Okay, pleasant. See that spongy. You can also have digestive aids. We also use it to alleviate any digestive discomfort. Okay, true ginger aid that can also come. This function can also come in as economic important displacement. Also have antioxidant property. Yes, because it can also help to reduce oxidative stress. Oxidative stress that people also use traditionally as medicine to also alleviate one discomfort or the other, one event like fever. So people can use it and also to believe that it will cure or it will immune them for malaria and other kind of illnesses. And also to have anti-inflammatory property. Anti-inflammatory property like can also be used as a pain relief. Okay, pain relief. So this what people need to know about this specimen. The two questions that are likely going to come is the observable feature. The observable feature is on board here. Okay, you can zoom and see the observable feature. I think I have how many here? 1, 2, 3, 4, 5, and 6. It's only asked to state 5 or 4 observable features. You have all of it here. How it functions, the storage order, as a vegetative propagation, the food source, flavor and aroma. So use it for digestive aid. There is problem with digestion. And also alleviate that discomfort. Okay, have antioxidant property for reducing oxidative stress. Traditional medicine also do using that belief. It can cure some 1 or 2 illnesses. And also have an anti-inflammatory property. It can also reduce or use as a painkiller. These are what we're discussing here on this specimen. So we go to specimen G quickly. Okay, specimen G is an onion bud. Onion bud is found on 1.5 leaves and the stem, as you can see in the diagram, in the picture shown in this screen. So we have its 1.5 leaf stem, is where an onion bud is found. Observable features. What are the things you observe when you assess the specimen? So the flesh is killed. Say very fresh cooking, the pasta plate that's on the bottom, and that become very stem from the top. Okay, so these are the things that you observe when you assess. Also some mark. Okay, some mark. The onion balls and around storage, and around storage of the onion bulb itself contains items of substance like food, water, and nutrients. One of the economic importance of onion bulb, just like ginger, both are used as what? Both are used as spices or food flavor because they give a pleasant aroma. They are used to spice up food. Okay, so you also can also deny the fact that just like ginger, do also have medicinal purpose, same as onion bulb. So you can also use it, also do have medicinal importance that some can also use it as an antioxidant to also ease or alleviate oxidative stress, just like the ginger. So the most important thing you must know about this particular onion bulb is that onion bulb is an underground storage organ. Okay, that contains what? Foods, water, and other essential nutrients. They are modified, formed from the modified leaf and the stem. Okay. Now, the means of vegetative propagation is specimen onion bulb is by body. You see young blood coming from so. I see a case where this particular specimen is drawn on the day of the exam. So when you are drawing the diagram, draw the size, let's look at the specific size of the diagram, compare with the size of the live diagram. Okay. You can see the diagram also you have a caption what I drawing maybe an onion bulb that is cut longitudinally placed there on the table, then you now start drawing based on what we are seeing. So know that this is also how to have enough hardware to just see it under. So when you draw, there are things also that you consider when you are drawing. Like you can see the diagram there. You draw and make sure it stays within the size of the diagram. You can also make sure you label accordingly to ensure that no marking be lost from the neatness of the label. So take notes. So they should, as a matter of fact, learn how to draw and remember and label accordingly. So the next specimen we will be looking at the specimen H. Okay, thank you.